So second speaker is JP Flores. He's from the UNC, and then his talk will be the hyperosmotic stress induces the complete rewiring of chromatin interactions. Look forward to your talk.
Cool. Yeah, thank you so much. There's a lot of you here, so I'm going to try not to black out. It's going to be great. Yeah, my name is JP. I use he, him pronouns. I'm over at UNC Chapel Hill. And in the first half of this talk today, I'll talk about 3D chromatin structure very generally and how we study that in lab. And in the second part, I'll talk about its role in the environmental stress response. But before I begin, I did want to say I love conferences like the Nabrit Conference because it brings together scholars from different career stages, fields, to showcase work from underrepresented scientists. And I love the activities like the professional development panels and the ones about identity. Right? And when talking about my identities, I'm a first-generation college student. I played college baseball, and I'm also a part-time bartender. So I'm a part-time bartender at this brewery just outside of Chapel Hill, North Carolina. And when I moved from Los Angeles, California to Chapel Hill, this was kind of like my second home, right? And for many reasons. One, the tight-knit community, but also the artwork that this brewery has on its labels. So for example, this one is named after the Taylor Swift song Blank Space. I love the rocket raccoons on this label, the UFO and the animals here. And there's also a label named after the Steely Dan song, Stole Away the Time, right? So there's that. One day I was bartending, and I was like, yo, one would be really cool is if I took the artwork out of all these labels and used it in a talk to talk about 3D chromatin structure. And that is exactly what I'll be doing today. So let's get into it. DNA, as we all know, is the building block of all living organisms. It can be represented as a sequence of letters. And one strand of DNA is about 3 billion letters long. Right? And what I think is really interesting is if you were to stretch out DNA from end to end, you'd find that it is about 2 meters in length. That is equivalent to 6 feet. Right? And when I first joined this lab, my background is in biochemistry, I was kind of like, wait, hold on. So you're telling me 6 feet of DNA has to fit into a cell nucleus that is about 6 microns in diameter. This totally blew my mind because that meant things like Harry Styles, 1.5 JPs would fit into a cell nucleus, right? And just to belabor the point, right, we have about 5 trillion cells in any given individual. And if you do the math, that means we have about 10 trillion meters of DNA in a person. And that is about 100 times the distance from the Earth to the sun, which is really interesting. Now, back in undergrad, I knew nothing about chromatin biology. Right? I was like, okay, chromatin in a nucleus, but it turns out it's not just random. It's not just like spaghetti in a bowl, right? It turns out that there's a lot of order inside the nucleus. So, for example, within the nucleus we have chromosomal territories. Within that we have compartments. Within compartments we have these things called TADs, or topologically associating domains, synonymous with chromatin domains. And the basic unit of 3D chromatin structure are chromatin loops, which is a topic for today. What else do we know about the genome? Well, we know that genes are only 2% of the genome, and one might ask, what is the other 98% doing? And a lot of work in the field has shown that over 30% of the genome is regulatory. But what exactly do I mean by that? So I'm going to pull one of my favorite steel string characters, this banana guy. He's going to spit out this line of DNA for me. So let's pretend like this is linear DNA. And in this region, we have a gene, right? And upstream of that, we have a regulatory region called a promoter. So what the promoter does is it serves as a loading dock for transcription factors to come in and basically recruit other proteins like RNA polymerase II to transcribe DNA to RNA. Elsewhere in the genome, we have these regions called enhancers. And they're pretty intuitive, right? What they do is they enhance the transcription of their target genes and promoters. The interesting thing about enhancers is that they can be tens or hundreds of base pairs away from the target gene or promoter, or they can be hundreds of thousands or millions of base pairs away from its target gene or promoter. And it's still able to recruit these transcription factors and help recruit RNA polymerase II to transcribe DNA to RNA. So how is that possible? One mechanism that's well known in the field is this mechanism called loop extrusion. And in this mechanism, what happens is you'll have 2 CTCF proteins, one bound to the enhancer, one bound to the gene. And then you have another player in this. It's a protein complex called cohesin that kind of goes around and scans DNA. And because it's ring-shaped, it's able to extrude DNA through its ring-shaped complex until it hits those CTCF blocks. and that's how a chromatin loop is formed, through loop extrusion. All right, it's bringing an enhancer and a gene close together in 3D space. Cool, so how do we study this in lab? Before I talk about how we do this at the molecular level, I have one more analogy that I want to bring up. This one is using Club Penguin. If you are unfamiliar with Club Penguin, it is a massively multiplayer online game that was really intense when I was 9 years old. Let's say you're playing Club Penguin, right? And I'm going to bring all the station characters into this. Let's say I wanted to track all the pairwise interactions of all these characters with each other, right? Remember, enhancers and genes, it was pairwise. There's one enhancer, one gene. That's obviously not the case, but that was just a toy model. But let's just pretend here. Let's say I wanted to track all these different pairwise interactions. One thing I would do is I would just sit here and watch all of them talk to each other, right? But as each one interacted with each other, I'd be taking pictures. So I'm taking snapshots of when these characters were interacting. The next thing I would do is I would create a matrix, right? So on the X and Y here, we have all the different steel string characters. It's the same one, so rabbit on a sailboat, rabbit on a sailboat. Here we have a heat map legend that indicates the number of interactions, 5 being the max in this toy example. And this diagonal you see is just representing self-interactions. So for example, pig is always interacting with pig because it's itself, right? So what we do is we just track the number of interactions. So let's say we had a banana guy interacting with rabbit on a sailboat once. We do this for 2 interactions, 3 interactions, 4, and 5, and that's how we track, that is the analogy that I'm going to use for how we track things in the genome. And there's always values to them as well. All right, so how do we do this? We perform a technique called in situ Hi-C. So how does this actually work scientifically? Right? So let's say that we're looking at this orange and purple region together, purple and orange region, and they're close together in 3D space. The first thing we would do is take a picture, right? So we add a chemical like formaldehyde, cross-link the cell, and this glues everything in its place, stops everything in its place. We would then add a restriction enzyme. So what this does is this makes random cuts all over DNA. We would then dilute away fragments that weren't glued together. There's a proximity ligation step where things that were close together, we can ligate them together. We then reverse cross-link or take off the glue. And we then have this chimeric fragment that we can sequence and align to our genome of interest. And this is what a Hi-C matrix would look like, right? So it's the same concept as the toy model. We have chromosome 2 on the X, chromosome 2 on the Y. We see a lot of color here, indicating a lot of interactions. And what this is saying is basically at this position, 62.4 megabases on chromosome 2, and at this position, 62.8 megabases on chromosome 2, these 2 regions are interacting. Cool. And the way that we view it in our lab is we actually turn it on its side and have it in this rectangular format rather than a square. Cool. So now we're all Hi-C experts, right? in that 5 minutes. Cool. So in this next part of the talk, I'll be talking about the role of 3D chromatin structure in response to environmental stress. So several studies have shown 3D chromatin changes in response to environmental stressors like hypoxia and heat shock. Today, I'll be talking about hyperosmotic shock. So why hyperosmotic stress? Well, it turns out in different organs like the guts, liver, and even the eye, cells are undergoing constant states of hyperosmotic stress. And it has to mount a response to that, right? And if that response goes awry, that can lead to diseases such as type 1 diabetes or cancer. So there was one paper in the field that looked at hyperosmotic shock in breast cancer cells. And what they saw is a loss of loop extrusion machinery. So the CTCF proteins and cohesin, RAD21 is a subunit of cohesin. You can see that after treatment of a hyperosmotic stressor like NaCl, you basically lose all of that machinery. 5 minutes? Okay. And what we did is we took the Hi-C data in this paper and looked, and I'll talk about what these are. These are aggregate peak analyses plots. I'll cover these in the next few slides. But we were able to show that after stress, we actually get a loss of chromatin loops in this system. So this is a really important paper in my thesis. This came out of Jennifer Lippincott-Schwartz's lab over at Janelia. It was published in Nature Cell Bio, where they developed this technique to visualize regions of accessible chromatin. And when I first saw this paper, me and my advisor were like, oh, okay, cool. So they're treating cells with a hyperosmotic stressor, HEC293 cells, with this hyperosmotic stressor called sorbitol. And in the untreated condition, there were regions of open chromatin, but after treatment, they saw new regions of open chromatin come up. So we were thinking, oh, maybe 3D chromatin is changing. So what we did is we were like, okay, did the Amat paper do this, the paper I just showed you? They did not. It was because they had lower sequencing depth, but we decided to just look at their Hi-C data and look at potential gained regions, and we saw a gain in chromatin looping after hyperosmotic stress. So we went ahead and were like, let's contact this lab and try to collaborate. And like a true scientist, we had the meeting. I was panicking because I was like, oh man, this is my thesis now, and we went along with it. So we took their cells, HEC293 cells, and we treated them with sorbitol after 1 hour. So you can see all these beautiful chromatin interactions. These triangles are an example of a domain. And it turns out after you treat them with sorbitol, you get this massive loss of chromatin loops and domains under hyperosmotic stress. This is one example region on chromosome 1. We also see this loss happening genome-wide, but we also see a gain of a couple of hundred loops, which suggests that this is a genome-wide nuclear rewiring. So again, here's another example region on chromosome 1. You have all this beautiful chromatin structure, and you see that loss after sorbitol treatment. But you also see the gain of new chromatin interactions. These are also incredibly well-powered, by the way. This is like, very well-powered. I can show, I have appendix slides for that if you want to see it. So we were really interested in figuring out, okay, what are these chromatin interactions? Could this be upregulating genes to adapt to this hyperosmotic stress, for example? And real quick, we wanted to keep digging into this Hi-C data. So here is I'm going to explain what the aggregate peak analyses are. What we do in these plots is we take all the loops that were found in like an untreated condition and pile them all up. And we do the same thing in the treated condition. So you can see that the loops that were present in the untreated condition are not present after treatment. We do the same thing for our gained loops. So loops that were present in our treated condition, we know that they're not present in our control condition. And this shows a true differential across treatments. So I don't have a lot of time, which is why I only stuck to one figure. But this part of the paper really showed that that hyperosmotic stress really does induce loss of chromatin loops and chromatin structure. These loops appear to be CTCF cohesion independent mechanisms, and the sorbitol-induced loops exhibit a more punctate pattern in Hi-C maps, right? So like this loop looks very different from this loop, and we have a lot of reasons or a lot of ideas for why that is. And if you want to talk about it, we can. I need to advance through this. Cool. So I promise I didn't just do Hi-C. This is a almost finished story where we've mapped chromatin interactions. We also are timing the formation of these gain loops using DNA fish, which is a great orthogonal method to Hi-C. We also have cut and run and attack-seq libraries. So cut and run maps protein DNA interactions, attack-seq regions of open chromatin. And we're also doing a time course of RNA-seq to really unravel the mechanisms of this new chromatin formation. And because, and every computational person has their wet lab buddies. So these are all my wet lab buddies in lab. They're phenomenal. And I didn't want to leave today without talking about different DEI initiatives that we're on. So a couple of years ago, Tina Termini over at Fred Hutch created this 100 Inspiring Hispanic Latinx Scientists in America list. Doris and I, they're prep scholars, Doris, Crystal, and I, they're prep scholars at UNC. We decided to reach out and say, let's expand this list. So this is a secret, please don't tell anyone, but if you know anyone that is Hispanic Latinx and is a scientist, please let me know, because we are currently gathering all that information right now. So we have that going. I also have a podcast where I interview scientists from all over the world and ask them how they got there. So Bill Nye, Francis Collins, Carolyn Bertozzi, people here, Freeman Hrabowski, Mike Summers, I think Dr. Tolbert talked about him yesterday. It won a Spotify award in 21, was Podcast of the Week two weeks ago. And I'm also on this project where I designed and taught a class about promoting inclusivity in the research lab environment at UNC. And I've contacted people at Stanford and all these other, like Michigan, for example, and we're trying to figure out, how can we expand this networking curriculum. And with that is the end of my presentation. I want to thank my lab, committee, department, funding, and if you want to reach out, please email, or this is my social media. Thank you.
Even before I ask a question, here we go.
Actually, it's more of a comment.
Oh, yeah,
Joking.
yeah, for sure.
This was the most amazing and beautiful presentation I have ever seen in my life.
Before this, I was really stressed out.
I was like, I don't know how the crowd's going to take it.
Good. It's amazing. Beautiful talk. Amazing. I mean, all the things that we chatted about, it really came out. I just want to echo that. I do have a scientific question. How dynamic is the genome and its ability to reorganize under non-stress conditions?
Incredibly dynamic. That is the issue with these genomic studies, right? Is like, that's why we're doing all these time courses, is that Nature Cell Bio paper, they saw chromatin changes within seconds. And when you're thinking about a technique like Hi-C, it's kind of like, well, if we cross-link at 5 minutes, how do we know that that's actually going to capture what we want? So we're also supplementing this with like live cell imaging. It's going to be a bigger paper than what I would like, but I'm also just super interested in chromatin dynamics and just want to figure out what's going on. So it's been awesome.
Hi, really great talk. I just had a question. Do you see the same effects when you treat with NaCl as with sorbitol?
Yep, we see it with NaCl, sorbitol, sucrose, KCl. It's like across even cell types that we see these loops form.
Okay, one last question.
Thank you for a great talk. I was wondering if you could say something about if it's known, the mechanism connecting hyperosmotic stress to the chromatin changes.
Say that one more time.
The mechanistic connection from hyperosmotic stress to the changes in the chromatin.
Like, what do I think hyperosmotic stress is doing to the chromatin to cause those interactions?
Yeah, like the chain of cause and effect.
Oh, yeah. So like, yeah, I don't know the answer to this, but hyperosmotic stress does a lot to a cell. For example, like we see cell shrinkage in microscopy studies. And I think it's really hard to say, oh, everything that's happening in the nucleus is the reason why the cell is able to adapt to that. I think that is like not necessarily true. I think a lot is happening to a cell under hyperosmotic stress. Yeah, this isn't the direct link, but we do think it's a contributor to its response to that.
All right, thank you.